Okay, we are ready to get started. Welcome everyone to the Akari Therapeutics Corporate Update webcast. As a brief reminder, all participants are currently in a listen-only mode. Following the presentation, there will be a question and answer session. Note that this webcast is being recorded at the company's request, and a replay will be made available on the company's website following the end of the event. At this time, I'd like to remind our listeners that remarks made during this webcast may state management's intentions, beliefs, expectations, or future projections. These are forward-looking statements and involve risks and uncertainties. Forward-looking statements on this call are made pursuant to the safe harbor provisions of the federal securities laws and are based on Akari Therapeutics' current expectations, and actual results could differ materially. As a result, you should not place undue reliance on any forward-looking statements. Some of the factors that could cause actual results to differ materially from these contemplated by such forward-looking statements are discussed in the periodic reports Akari Therapeutics files with the Securities and Exchange Commission. These documents are available in the Investors section of the company's website and on the Securities and Exchange Commission's website, and we encourage you to review these documents carefully. So joining on today's call from the Akari Therapeutics leadership team are Abizar Gaslightwala. He is President and Chief Executive Officer, and Dr. Satya Mitra. He is Head of Oncology. I would now like to turn the call over to Abizar. Please proceed.
Great. Well, thank you, Janine, and welcome everyone. We thank you for joining us today. Go to the next slide, please. So I'm just going to give you a brief overview of what we're going to talk about today. I'll just give some opening remarks and a corporate overview for Akari. Then we'll also get into the payload overview for ADCs, kind of what the landscape looks like and how we are a little bit different, but then we're going to kind of roll that out a little bit more with Dr. Mitra. He's going to walk through some of the really interesting data that was presented at SITC, the Society for Immunotherapy of Cancer, about a week and a half ago in Washington, D.C., where he'll do a deep dive into our novel payload and talk about what's unique about it and interest to focus on our splicing targeting payload and the ADC data that we presented. And then I'll conclude with some follow-up steps on our lead asset, AKTX 101, and where we're going in terms of development plans and next steps, and we'll do some Q&A. And again, as a reminder for Q&A, you'll be able to submit your questions through the webcast, and our IR team will be able to field those to us. Go to the next slide, please. So I just want to give a high-level overview. Many of you might be familiar with this already, but just as a brief orientation, within oncology, which continues to be the number one therapeutic area in the pharma biotech sector, antibody-drug conjugates, or ADCs, continue to be the most popular and exciting modality to treat cancer patients. These have continued to be prominent in the news, as you've probably heard and seen. And as an example, there are over $6 $1 billion, what we call blockbuster products already that are ADCs, across a number of different cancer indications, whether they're solid tumors or blood cancers. And this class is rapidly growing and accelerating as well, with new products entering every day. And this market is expected to surpass over $30 billion shortly. And as we look at these ADCs, you see some of these at the bottom, some of the names you might be familiar with. You can see the different types of tumors they go after. You can see the respective sales just in 2024, cumulative sales worldwide, and these are continuing to accelerate. What's interesting, when you look at the payload that's used on all these blockbuster ADCs, and importantly on the right, which is all the ADCs in early to mid-stage development, you see two payload classes dominating: DNA damaging agents and microtubule inhibitors. And those are two kind of biological mechanisms how these payloads work, which are very similar how chemotherapy, traditional chemotherapy works. So yes, some of these ADCs use targeted chemotherapy, maybe different versions of the older ones, but these are the two payload classes that over 95% of all ADCs are using. And so although there's been tremendous success with these two payload classes, which again are versions of chemotherapy, and they've done a great job with the current ADC set, we want to walk you through why it's important to diversify this payload approach, because as good as these ADCs have become, there's more opportunity to go beyond the current efficacy and safety profiles we see. So let me walk you through why that is. Can we go to the next slide? So in terms of Akari, we're advancing ADCs beyond today's efficacy and safety limitations you see today with urgency and efficiency. So getting to that first point, payload innovation for ADCs. Why do we need that? Well, as good as those ADCs have been that we just walked through, there are limitations. Let's take, for example, an ADC class called TROP2. It's a target on many different solid tumors. And when you look at some of the current ADCs approved in the US market for TROP2s, what you see is some interesting stats. You see the median progression-free survival, which basically is the amount of time someone as a cancer patient goes without their tumor progressing. That means 50% of patients get less of a response. And this response on average for these current approved ADCs is only about 5.5 to 7 months. So imagine roughly about 6 months of benefit you get with these ADCs. Now, it's better than where these patients were before, but 6 months doesn't seem to really move the needle for many patients and their families. We also see tremendous side effects still with these ADCs. As I mentioned, many of these ADCs use chemotherapy-like payloads, so you're going to expect chemotherapy-like toxicities. And one of the more important things we look at, both for a patient and a physician, is dosing interruptions. When a patient actually has to stop their dosing of a product because their toxicity is too bad, well, that's bad because that means you're not treating the cancer. So when you look at the, again, current TROP2 ADCs, anywhere from 22 to 66% of these ADCs had to be dose interrupted in the clinical trials while they were dosing patients. And that's a best class scenario because clinical trials are just an objective experiment. So imagine, again, the tolerability and safety issues. So when you look across efficacy and safety, there's clearly, despite progress by these current ADCs, there's clearly opportunity to improve on both. And that's what makes us excited about Akari's PH1 splicing modulating payload. It is totally different than those chemotherapies we've heard about and talk about. It has a 1-2 punch, and we'll speak more about that in a second. It is cytotoxic, which means it kills the cancer cell it's targeted. But more importantly, it has a strong immuno-oncology action, a dual action for the payload. And we know that immuno-oncology, the immune system, is the best way to actually attack cancer. And we have a payload that does that in full totality. And we're going to talk about that data and what that was presented at SITC. So we want you to anticipate that very soon. We also know from our preclinical data that this payload PH1 is superior, what we've seen in preclinical testing, to some of the current ADC payloads. And we have a number of data, and we'll walk through some of that as well in terms of some of that data that gives us conviction and confidence in this payload's performance. We've also seen in our initial safety testing that the payload for our ADC, this PH1, has a differentiated safety profile. We don't see some of the safety events and issues that we see with the current ADCs, and that gives us a lot of encouragement that hopefully we can improve that statistic around dose interruptions, and we can see better dose continuation, which means you're treating the cancer more readily. So this is the reason why we think payload innovation is needed in the ADC market to fully unlock the potential, and we have a great solution with our PH1 splicing modulating payload, which you'll hear more about. Well, what does that payload mean? Now that Akari has a first-in-class, we think best-in-class payload, we can actually start applying that. And we have our lead asset, AKTX-101. It's actually going after TROP2 as well as a target. And we're going to speak more about that at the end of the presentation. We believe this payload can be leveraged not just for AKTX-101, but across many other ADCs to create a pipeline of ADCs. So we think there is a pipeline in this payload opportunity. And we've already demonstrated in preclinical experiments, and you'll see some of this shortly, we think this payload is super active in many areas. We actually filed some patents early this year because of that. We believe we're active in certain cancer mutations, or oncogenic drivers are what they're called, that are pretty bad in certain cancer types. We actually find our payload is very effective against those. We filed a patent around that. We also know our payload is very active in immune activation. We filed some patents around that in the fourth quarter, and you'll be seeing that data that was presented and part of that patent filing as well shortly. And the last piece, I want to kind of differentiate Akari maybe from other biotechs. How do we think about ourselves in terms of not only this innovation, but how we're bringing this innovation to market? And we're doing it with urgency of speed and efficiency of capital. And how are we doing that? Well, we're going to be using a lot of partnerships to incentivize speed to get to the clinic. And we'll hear more about that on the 101 program shortly. Because we think that partnerships, risk shares, and opportunities to get people with the same incentives and alignment to deliver results is more important than just paying vendors to do work. That also allows us to defer capital outlays until we get to value inflection points. As we progress our pipeline, not only do we benefit, but our partners benefit. And that's how it allows us to be more efficient with our capital. It allows us to advance our preclinical assets like AKTX-101 to the clinic at much lower cost than are traditionally seen from ADC development, and really allows value inflection for Akari. If we can get these assets quicker into the clinic, we could get a valuation for Akari that could approach what clinical stage ADC companies are today pending we generate positive phase 1 data into the clinic soon. So that's the Akari story. We're really excited about the payload innovation, but how we're actually executing against that payload innovation to bring this new type of ADC to patients, hopefully in the future. We go to the next slide. And with that, I want to transition to Satya. Dr. Mitra is our head of oncology, and he will walk you through the exciting data around the payload, set up some background, as well as walk you through the SITC data that we just presented with great anticipation and excitement. Thank you.
Thanks, Abiza. Next slide, please. So, Abiza talked about how exciting the ADC field is, and my first slide is about the limitations of ADC. So, don't get me wrong, it's the fastest growing class of the modality which has upended traditional chemotherapy and upended many targeted chemotherapies on the way to success. So, but as is often the case with something, a modality that leapfrogs very quickly, it creates a vacuum in its space. So, since 2013, HER2 positive breast cancer and gastric cancer patients were receiving the drug Kadcyla, and many years later, this was replaced by a drug that performed better, Enhertu. Then the question immediately rises, what do you do in a post-Enhertu setting? Is the target still relevant? Do the cancer patients still express HER2? Do we give HER2 ADCs with different payloads? Well, from early studies seem to suggest that the resistance is due to the payload. So you can follow a HER2 ADC with another HER2 ADC of a different payload class, and you can still expect activity. But if you follow the same, a different target, ADC against a different target, you have, with the same payload or the same payload class, you may not capture that activity. So this immediately posed an important question. Where does resistance come from? Where are the mutations that give rise to resistance? And you can see in various patient pools, there are mutations in topoisomerase that make it refractory not only to the payload that ADC with the same payload that it is being treated with, but also potentially to the payload class. So anyone who's been treated with an irinotecan version, the mutation makes it refractory to deruxtecan and reduces the binding affinity to something like exatecan. And there's about 60 such topoisomerases coming through the ranks in clinical studies. So this poses a big challenge in oncology. How do you sequence ADC after ADC? The other problem is when you look at the resistance per se, and you look at the toxicity profiles. One of the toxicity problems is that there is toxicity with a payload class. So irrespective of whether you treat with any version of irinotecan, you have nausea, vomiting, diarrhea, you have gut toxicity associated with that payload class. You're not going to walk away from that. You get less or more of that toxicity as you engineer incremental changes around the payload. You tweak a linker here or there, or you make small changes about the payload, you're still stuck with the toxicity of the payload class. So how does one deal with that? You have to change the game. You can't play by the same rules and expect to win. So this screams out for the field to generate different payloads, and payloads of a different class altogether, different mechanism of action altogether. So what we did is we started working on a novel payload class, and we asked ourselves the question, what are some of these hallmarks that we should capture? One important hallmark is cytotoxicity itself. We should have the ability to kill cancer cells. In addition to that, we thought that there was no better modality than activation of the immune system. And looking at the success of checkpoint inhibitors across the spectrum in multiple indications where it brought out efficacy, unlocked efficacy of 30% of all patients, we decided this is our Godzilla. We need to get this in our payloads. So we engineered and looked for biologies that were compatible with this philosophy, and we discovered splicing at the heart of intersection of both these two, cytotoxicity as well as activation of the immune system. Then the problem with trailblazing and being first in class is you don't know what problems to expect. And all the patients that are developing resistance to either microtubule inhibitor class or topoisomerase inhibitor class, they have a common problem, and that is overexpression of efflux transporters. You do your best to dose the patients, get the drug levels to the extent that it causes tumor regression, but then the cancer cells adapt by overexpressing these drug efflux pumps and pump these payloads out of the cancer cell. By being second, what we can do is engineer our payloads and look for payloads that are refractory to drug efflux pumps. So we tackle this problem by basically stepping outside the box and going into splicing biology. Next slide, please. So what is splicing? You've heard the central dogma, DNA to RNA to protein, and in that, in a very key step is that of splicing. Splicing impacts about 80% of all human genes that have non-coding regions in them. And these are precisely spliced out before functional proteins are made. If these proteins are not excised properly, then there is a gate check mechanism called nonsense-mediated decay, which takes care of the proteins that are going to be misfolded or misspliced. And as a result of that, these never go on to make functional protein. What we do with a splicing modulator payload, ADC, is that we throw a wrench in these works. By throwing this wrench, you are impacting hundreds of proteins that are vital to the cancer cell, and therein is your cytotoxic mechanism. Some of these proteins will misfold and gum up the endoplasmic reticulum and the protein factories, and that results in death by unfolded protein response or ER stress. Finally, if by some miracle the cancer cell does survive this onslaught, then these misfolded proteins, these unnatural proteins called neopeptides, they are presented on the cancer cell surface, and these look like foreign proteins to the immune system. So they light up the cancer cell like Christmas lights, and this activates the immune system and kicks it into high gear. Next slide, please. So we've heard the story of every chemotherapy or every ADC payload basically saying, we kill cancer cells and creates a level of immunogenicity, or it releases new epitopes and activates the immune system. But the question is how much? How much purchase do you get when you tweak the biology towards production of these foreign proteins called neopeptides? So in this comparison, on the left, we're going to look at a payload which targets microtubule, and this is called DM4, and it's found in the folate receptor ADC Elahere. So we're going to look at how much neoepitopes that ADC, that payload makes, and compare it to ours, which is on the right. So this is a transcriptomic study. You're looking at all RNA species across the board in an unbiased manner. All the stripes through the middle in black reflect genes that are unchanged, and anything above that is increased, anything below that is decreased. So in the blue, we appreciate all the genes that are going to be depleted from the system and cause cytotoxicity. And you can find that there is a threefold increase in the number of depleted genes by our payload. So 199 versus 660. So what we see here is a more potent cytotoxic mechanism with respect to the DM4 payload from the splicing perspective. Now, if you look above and focus on the red dots, here we are asking a question: where are the proteins that are not found in the human proteome? So these are like foreign proteins, these are like new epitopes. And when you look at these sequences, you find that there is almost a tenfold increase in the payload-induced new epitopes. What that means for us is that while our payload is also cytotoxic and will create new epitopes by all the classic mechanisms that other chemotherapies and ADC payloads do, we will produce a different class of new epitopes of the frame shift category. So now let me talk you through how these two are different. New epitopes from cancer cells are generally something, a carbohydrate residue that is higher in cancer cells versus normal cells, or it is a property of two sequences coming together, and you have a 2 amino acid difference at the junction. But a splicing modulator causes a frame shift for all the amino acids downstream of that where you threw the wrench in the works. So it has the impact of creating hundreds of amino acids that are different. So what I'm telling you is that all new epitopes are not created equal. And this class of new epitopes, called frame shift new epitopes, which are induced by a payload, increase the antigenicity, and these basically kick the immune cells into high gear. Next slide, please. So this is one of the slides that we presented at SIPC. And here you can appreciate what this payload, how it changes the characteristics of the same antibody. So this is, we spoke about Kadcyla, right? Now we're going to go head to head against Kadcyla, plus minus anti-PD-1, in a colon cancer model which has HER2. So what we've done is we've treated these mice bearing tumors for a period of only 14 days with the ADCs, and we followed it for a period of 5 months to see tumor regression. And when you get tumor regression, you want to know the mechanisms behind it. If there is an immune mechanism, you want to know what that is. If you see different rates of complete regression, we want to understand why that is. So over to the right, you see all the individual tumor growth curves in our mice experiment. What we see is that our ADC, called TRAS-PH1, gives you nearly the same efficacy, captures the same efficacy of that of Kadcyla, and slows down tumor growth. But in addition to that, it causes a small rate of complete regression, about 11% of the mice. And this results in the tumors going away completely. That's what we call complete regression. Now, in this experiment, the checkpoint inhibitor, anti-PD-1, worked really well and cured 33%, 1/3 of the mice of their tumors. And even in that backdrop, our ADC combined very well with checkpoint inhibitors to unlock further efficacy. So if you look at our combination, TRAS-PH1 plus MPD1, and compare that to Kadcyla, we find that there is a near doubling of complete regressions. 14 out of 19, 74% versus 42%. And this was behind some of the efficacy that we observed. So now let us find out why we saw this efficacy by doing something called immunoprofiling, looking at the immune cells when the cancer regressions were happening. And we find that each arm was associated with a different immune mechanism. Next slide, please. So our single agent ADC did something which was really unexpected. As expected, we induced the frame shift neopeptides. That's the mechanism of the payload. But as a result of increased neoepitopes, we found all the antigen-presenting cells, the macrophages, responding and getting activated. These then presented these antigens to B cells, and B cells made anti-neoepitope antibodies. That was the primary mechanism in the class PH1 alone arm. We saw some changes, and these were anti-neoepitope based changes. There were no surprises in the anti-PD-1 arm. The anti-PD-1 arm had an effect on T cells. It unlocked and unleashed their activity. And that is pretty much expected from anti-PD-1. But when we put anti-PD-1 and TRAS-PH1 together, we not only saw both the individual activities of anti-PD-1 and TRAS-PH1, we saw something in addition to that. And that is, in our jargon, is what we call true synergy. We saw activation of a kind of lymphocyte called gamma delta T cells. And gamma delta T cells are the one of two lymphocytes, other than natural killer cells, that are capable of finding tumors and killing them without the need for antigen-presenting cells. So they are epitope independent. There is an entire industry of people who make gamma delta T cells, train them, and reintroduce them back into cancers to unlock efficacy. And we are getting those gamma delta T cells activated downstream of our payload. So basically, I want to summarize this section by telling you, in terms of getting differentiated efficacy and differentiated safety, one has to venture out of the box to get outside the safety zone of the conventional payloads. You have to try something different. You cannot get the same activity. You cannot expect different things by doing the same kind of sequence. Incremental changes are not going to bring about unleash a totally different spectrum of efficacy or a different spectrum of safety. And by working on this payload over the years, we have found that this payload, irrespective of which target we hook this payload onto, we capture the immune phenotype as well as the cytotoxic phenotype. So we are really excited to try this in multiple cases. So we presented this data at SITC, and it was received very well. I mean, at the talk, there was the first person at the mic said, just spontaneously, this is ingenious, and then followed with a tough question. You would expect that. We got an overwhelming response at our poster session. All the usual suspects from pharma came to our poster, looked at it, something new and novel on the horizon. We excited academics as well as MDs alike. We had people from academia offering ideas and suggestions by the minute. And they just couldn't help themselves. They're going on and on with these suggestions and making mental notes. And KOLs, very reputed KOLs who've done 50 plus clinical trials, when are we going to get our hands on this ADC, on this payload to try this for our patients? And all I could say is soon, soon, soon. So anyway, luckily I didn't have an answer for them then, but Abizar has an answer for you now. So I'll hand it back to you, Abizar.
Great. Thank you, Satya. That's a good segue for what I'm going to go through. Let's just stay here on this slide for a second. I just want to recap, Satya. Thank you for that really comprehensive overview of the history of ADC payloads, the limitations, and the whole design principles of you and the R&D team of why you came up with something to go after RNA splicing biology and the payload that's been developed through years of testing and these fantastic results. I think if I were to take a step back, I would say these results are unprecedented for any ADC payload, because in any of the literature you look at, no payload has demonstrated this type of immuno-oncology effect. It's very unique. This is charting new ground, which is why the SITC organizers wanted us to have a main oral presentation and a poster. And what I think is really interesting, just to kind of, I'm not an immunology expert, but when I look at it, I think of these as two puzzle pieces. When you think about our ADC payload and what it does versus the checkpoint inhibitors, which are the most successful class of agents in oncology at $50 billion plus and growing, our agent activates this innate kind of immune system, which is one type, and the antibodies, the humoral B cell antibodies as well. And then the checkpoint inhibitors are the T cell. So when we marry them all together, we're getting this really unique response of innate, adaptive, which are T cells. Innate's the macrophages and neutrophils. The adaptive is the T cells, and the humoral is the B cells. I don't think any payload with any immunotherapy has ever demonstrated that kind of pleiotropic broad-based effect, which gives us continued excitement around we have something novel and how we want to progress it. So thank you for putting that in really simple terms for us to understand. Now, the question is, how are we going to take this in? As Satya said, into clinic and start demonstrating these results. As Satya said, this payload dynamics can be generalized to any antigen target. Hence why we're really excited. The data that Satya just presented was in a HER2 antibody that we put our payload on. That was a test prototype. Let's go to the next step and talk about AKTX 101. This is our TROP2 directed ADC. We have a lot of good translational data preclinically that gives us good insights into where we want to go. And what we want to do is we actually want to take this product now and do the final stages of manufacturing, final manufacturing and scale up, and what we call our toxicology final safety studies. We've done preliminary studies, and that is at the last step before you enter human clinical trials. These are the big steps. These mean that you have a really good molecule and you're ready to go prime time in a sense. So that's what we're starting. We call them clinical trial enabling activities, or what your IND enabling. That's a filing with the FDA to kind of start clinical trials. These are the activities you see on the top, the CMC and non-clinical, based on its manufacturing and final safety studies. And then that will lead into us hopefully starting this phase 1 initial first in human, that's FIH study in late 26, early 27. And our target area where we want to go into in particular, we would do this first in human and then potentially expand this into a really specific tumor type. And that specific tumor type is urothelial or bladder cancer in the second line setting. So what I want to kind of emphasize are a couple points. If you remember the beginning about what Akari is, we're not only about payload innovation, we're also in a sense trying to rework the biotech model innovation around ADCs. We want to get there in a very accelerated timeline. If you were to just ask other ADC companies, this is a pretty accelerated timeline to get to where we are now from a lead product candidate to actually starting a clinical trial within a year. But we're also doing it in a way where we're bringing partners in to help us defer capital outlays and do this in a very efficient way relative to the traditional model of how products are brought into the clinic. So we're excited that we're not only bringing innovation on the ADC side, we're also just trying to innovate how we actually, on the business side, how we progress things in different ways with risk sharing, with partnerships, with shared incentives, and capital efficient ways as well. And so we're really excited about this kind of process to get to the first in human trial specifically. So with that, let's go to the next slide. I'll just kind of summarize. So I think as you heard, we'll tell you again, why the need for novel payloads. Well, as you heard, as I mentioned, and as Satya really talked in detail, when you look at the current ADCs, as good as they are, there are limitations both on the efficacy and safety side. And we use the TROP2 ADC as an example in terms of the efficacy limitations and safety tolerability. This is true of other antigen targets, other antibodies, and those payloads, which are only two classes. And that leads us to why we have a splicing modulating payload, and what we put onto AKTX-101, our lead molecule directed against TROP2. We think there's continued unmet need in this TROP2 expressing cancers. And as I mentioned, maybe early in the slide, we talked about urothelial second line. Well, the reason we're thinking of second line is exactly the reason that Satya mentioned, because many of the patients in the front line have developed resistance or lost efficacy to the first line ADC, which uses a microtubule inhibitor. So exactly the reason we're seeing Satya talk about that theoretically is playing out in the market, and we see a growing pool of these refractory bladder cancer patients. We think that's a unique opportunity for us to go after. But we're not done there. We think TROP2 has a lot of legs. We know we could potentially go in gastric cancer. We have some really compelling data. And lung and breast are also known as potential strong areas that we could take our ADC into as well. And that's based on the robust efficacy we've had. So a lot of broad potential for our lead ADC. And that timeline I talked about to get to the first in human clinical trial data, we'd like to hopefully get more in the 12 versus 15, but we think that's the window, and that's on a very accelerated time frame in a capital efficient way. And I think the take home is ultimately we get into the clinic, we prove some of this pre-clin data in a human clinical trial setting. We think that is a really attractive value inflection for Akari that gets us into a different space comparable to other early stage clinical ADC companies. So with that, I really want to thank you for your time today, and we're going to open it up for questions and Q&A. And Janine from JTC will help us navigate that.
Great. Okay, so we are ready for the Q&A. To our audience, if you have a question, you can click on the Q&A button at the bottom of your screen, type in your question, and we will get to as many as time allows. All right, so our first question is from James Malloy. He's a research analyst with AGP. There's three questions here, or three parts to it. I'm going to read all of it, and then we can break it down if you want, because I think it's important to put it all together.
Yeah.
So the question comes in: how would you characterize the current partnership environment? Are there any partners currently in the data room or under NDA? And there's been some recent high-profile M&A in the oncology space. J&J and Hyeda and for $3 billion, Genmab and MRUS for $8 billion, and has this activity increased interest around AKTX's portfolio?
Sure. So I think I remember the second and third. Can you remind me the first? Sorry, the first one was?
Sure. How would you characterize the current partnership environment?
Yeah, so I think the partnership environment continues to evolve. I think you're seeing a variety of different partnerships. You're seeing outright licensing and acquisition of molecules. You're seeing company acquisitions. The Genmab one was mentioned as well. So we think there's actually increasing deal flow and activity. Maybe it gets to the third part of the question. I think in particular, you see ADCs in oncology a still bright spot in the biotech sector, which gives us a lot of excitement on our plan to move forward. You know, you saw a lot of ADC deals in the first half of the year. Just recently, in the last 4 to 6 weeks, if not broader, you've seen a number of deals on the ADC, whether they're financing, whether they're acquisitions or licensing by big pharma or not. I think it continues to show the excitement around targeted therapies, ADCs in particular. So we're pretty encouraged. I think in the second part, obviously, you cannot disclose confidential information around partnership discussions we have. What I can say is we enter a lot of both non-confidential, and we do have a number of confidential discussions we're having with potential pharma partners. Obviously, you can't give any more details than that, but we are active. And I would say the reason we're active, and you can imagine how many inbound some of these pharma companies get. It's the splicing modulating payload. What Satya just talked about, that is stopping power. That gets people to look at it again, and that's why people talk to us both on a non-con and confidential basis, to explore more about what does Akari have and why does it look different, and wow, this does feel different. And these aren't companies that are new to ADCs. These are companies that have experience with ADCs. So the fact that we're getting our foot in the door and having these conversations, again, speaks to the uniqueness and novelty and innovation on this payload. And I think if I were to comment on some of the deals, I think you look at the Genmab deal, obviously, but that was Maris was a bispecific, I believe, a little bit different technology, again, around targeted therapies in oncology. But I'll just mention, just Genmab in general, some of the bets they've made may not be playing out. And I'm not saying they made a wrong bet, but just, the Profound Bio acquisition they did a couple of years ago, they've discontinued 2 out of 3 of those ADCs. So I think even the thesis on how some of these traditional ADCs have played out, either due to the limitations that they mentioned or competitive dynamics of competing against the same types of ADCs, some companies are finding they need to change the playbook a little. And we think that's exactly a good segue for why our novel payload is changing the playbook for ADCs and gives us excitement. I think I got them all. Did I get all of them?
I think you did. Yeah, great job. Okay. Our next question is, can you speak to the unmet need in bladder cancer and the market opportunity?
Yeah, sure. I think it's a great question, and one might ask, and if you're familiar with the bladder cancer metastatic, so there's many different types of bladder cancer. There's, muscle invasive, non-muscle invasive. We're going to focus on the metastatic, which is the most severe unmet need, which essentially means the bladder cancer is spread and metastasized other parts of the body. That's where the prognosis is the worst for patients due to the overall tumor burden. In that setting, there has been significant advancements, and let me just kind of characterize. And after I give this, I'll have Satya to add any things I might miss on this. But when you think about this metastatic bladder cancer patient, therapies like cisplatin, which is a platinum-based therapy, effective but super toxic and limited efficacy, subject to resistance mechanisms, as Satya mentioned, for chemotherapy. That's been the frontline setting forever, using cisplatin, maybe combinations of cisplatin. And then, there's a bit of a revolution or change that happened in the metastatic bladder cancer market. In ADC, a product called PADCEV, enfortumab vedotin, uses a microtubule inhibitor, actually changed the landscape. And this is, again, I think, a success of ADCs, that you had a targeted agent like enfortumab vedotin that went into metastatic bladder cancer, showed some great data, both as a single agent in combination with a checkpoint inhibitor. And that is approved in the frontline setting. And PADCEV or enfortumab vedotin is more than $1 billion drug, and it's on its path to do even more. It's moving into earlier lines, what we call a neoadjuvant setting, which is where the cancer is a little less spread. And so it's a great success story. That being said, as great as the data is and what PADCEV and enfortumab vedotin has done, it targets an antigen called Nectin-4, which is different than the target we're going after. Both targets are highly expressed. Nectin-4 and TROP2 are very highly expressed in bladder cancer. As good as that data is, when you look at what we call, again, the median progression-free survival from their pivotal trial data on combination therapy, it's about 12 months. And that's a big advancement from where cisplatin was, which is about under 6 months. So it has made significant inroads. But think about 12 months for a cancer patient. When we say median, that means 50% of patients relapse less than 12 months and 50% relapse later than 12 months, hence median. So if you're a cancer patient, yeah, that's an advancement, but you're only talking about maybe less than a year for half of, you're 1 out of 2 that may be less than a year before your tumor is going to continue to progress. And that's not a great long-term prognosis. So we want to change that. So there is now a growing pool of patients that have taken PADCEV and other combination agents and are probably relapsing. And we know this pool is growing. And so that's exactly that target population. And it's similar to what other companies have done and strategies that you go after refractory populations with super high unmet need that are now refractory to an ADC, refractory to chemotherapy. And again, as Satya mentioned, going after those same patients with the same payload, like a microtubule inhibitor, probably not going to work because they become resistant. So that's exactly the perfect setting for us to take a splicing modulating payload like ours into. And we have a tremendous amount of preclinical data that's probably our strongest potentially in this setting for bladder cancer. So hopefully I gave some perspective, the unmet need and why we're excited. Satya, I just want to turn over to you to add additional thoughts and ideas on that.
Well, I think you captured most of it. The only thing I would add there is, the mechanism of resistance there, the reports are basically not. You have some amount of Nectin-4 loss, you have MDR overexpression. So it's a good thing that this payload has been optimized for non-binding to MDR. And it's against a different target, which overlaps in expression in about 80% of all urothelial cancers that express Nectin-4. So that being the case, you have now an opportunity to treat and patients where there is inflamed setting which responded to PD-1 and increase the neoepitopes in that setting. So to unlock further efficacy. So it's a good place to be in terms of frequency of high expressing target, and it's a good place to be in with respect to how hot the cancer setting is.
Yeah, that's a great additional point, Satya. Although we might look initially as a single agent in this population, it gives us optionality to actually bring forward the combination data with an anti-PD-1 or checkpoint to get the kind of results we saw preclinically. We'd love to replicate, because that could be the enduring efficacy we could see, and the unique kind of profile of our payload really comes to fashion. So again, that's exactly the vision we want to go. We want to go to these hot tumors, which are more immunoactive, and bladder cancer is one of these. So thank you for adding that additional perspective that builds on the SITC data.
Great. Next question comes from Aiden Husenoff. He's a research analyst with Ladenburg Thalmann. His question is, can you help us better understand the differentiation of AKTX-101 from Trodelvy? And given both are TROP2 targets, where do you think AKTX-101 presents the best opportunity?
Yeah, I'm going to give maybe a high level, and I'll ask Satya to go walk through the more details. So Trodelvy is a TROP2-directed ADC. It was the first approved. It's actually a pretty successful drug, but if you look at it, it's over $1 billion in sales. It's approved in the breast cancer setting, two major areas, triple-negative breast cancer, and then another subset of breast cancer called hormone receptor positive, HER2 negative. What Trodelvy is, it is a antibody against TROP2. It uses the payload called it's a topoisomerase 1 inhibitor. It's a cousin of irinotecan, which I think Satya mentioned has its own toxicity. So by definition, that payload will have very similar toxicities when you look at its package insert. It's got grade 3. So to get the differentiation point, it's got grade box warnings for neutropenia, which means low white blood cell counts, high infection rates. Potentially, it has high rates of diarrhea and some other GI issues. Those are irinotecan-like side effects. If you're familiar with that chemotherapy, it's a similar set of side effects for this payload. And there's also a linker that they use that I'll let Satya explain, that's different than the way we're using. So, it's around the payload is very different, has some of those historical legacies of topoisomerase 1s, and the linker strategy they're using, we would probably say, and others would agree, is maybe not optimal. That creates other side effects, but I'll let Satya maybe describe in more detail.
So there are a number of differences in our approach to Trodelvy. Trodelvy is basically the active moiety of irinotecan, SN38, hooked onto an HRS7 antibody via a CL2A linker. So this linker is acid-labile, and by the nature of the release of the payload is in the extracellular milieu. So whenever you go into a tumor, the environment is generally more acidic than the rest of the body, so it deploys there. But no matter how good you are at deploying the payload at the decided location, there is always leaching of payloads from the tumor and exposure to organs. So you get organ tox, and some of the gut tox is what Abhizer rightly pointed out. Our ADCs are non-cleavable, and they only deploy in context of the target that is expressed only on the cancer cells. So we are trying to partition the payload specifically to the cancer cell, and we've taken a lot of precaution to prevent this payload from causing systemic issues. And this is because of obviously of the immune phenotype. We want to make sure that there is no off-target effects. So you get a lot of off-target effects in Trodelvy, which is in tune with the bystander killing, but we have reduced that in order to get less immune flare-up. We do get bystander activity separately from the immune system. When we bring immune cells into the tumor, they take care of the heterogeneity problem. They target the tumor, the target non-expressing cells as well as the target expressing cells, both equally. So in that way, we counter the bystander problem in a different way through an immune bystander approach. So very differentiated.
So, and I'd maybe add a couple points. Thank you, Satya, for giving a little bit more technical scientific explanation. So essentially, we avoid collateral damage. The way we've engineered our linkers and payloads, we try to avoid that collateral damage. And we also, because of our payload, we actually cause immune activation in a different way, hence the kind of responses we get. And in you know, just to kind of bring that home, when you see, we've done some head-to-head work against Trodelvy, and in the gastric cancer model, just looking at the pure cytotoxicity, we have superiority to Trodelvy in a gastric cancer preclinical model. So that gives us a lot of conviction that, yes, this payload is super active and is capable of beating the lead TROP2 agent, at least in this preclinical model of gastric cancer.
Okay, our next question is from Viju Gupta. He's a translational scientist. I just lost my screen. There we go. At WashU. Viju says, great information. There are two questions. Number one, is your mouse model a sub-Q model or orthotopic? And number two, PH1 being related to statins, do you anticipate effects of cholesterol pathway?
I'll defer these to Satya. Why don't you go ahead and take them, Satya?
Sure. The first part of the question, we typically test our ADCs against tumors that are grown sub-Q, although we are not opposed to doing the odd orthotopic experiment. But in this case, whether we are doing urothelial models or gastric cancer models, these are typically sub-Q. The second question, whether PH1 being related to statins. So I don't see a lot of similarity in the molecules per se. The cholesterol molecules will have that per hydrocyclopentanophenolamine link system. This is very different from that. And even when we have done a non-human primate studies, we haven't seen any changes per se in cholesterol level. That seems to suggest that, you know, the cholesterol pathway may be important.
Our next question is, how does PH1 perform if it's used alone without PD-1? And in a clinical setting, would the sequencing of therapies affect its efficacy?
I'm going to let Satya maybe take that on, and I might add some following comments. So,
sorry, I lost audio there for a bit. Janine, can you repeat?
No problem. I'm happy to repeat it. So, how does PH1 perform if it's used alone without PD-1? And in a clinical setting, would the sequencing of therapies affect its efficacy?
That is actually a brilliant question, and that's front and center on everybody's mind, right? So the first part of the question, I'll try to break it up, is when you use this as a single agent, you do capture the cytotoxic efficacies, and you also see the effects on the innate immune system, because that's there in mice, in humans at baseline. So you see the effects on macrophages, you'll see the effects on neutrophils. So there will be some baseline killing and some baseline immune activation. You won't get the purchase of T and B cells, most likely. You won't get the activation of T cells without the anti-PD-1, but you do get activation of B cells and anti-neoepitope antibodies, even in the absence of PD-1. So that was behind the mechanism, if you remember, in the TRAS PH1 story. So we do get some activity. We do get long-term activity that we have seen in our HER2 models, which are xenographed. So by xenograph means we use nude mice there to allow the human tumors to grow out, and they don't have an immune system. So in those experiments, we do see very good killing, and it is in one of these experiments that we performed equally or slightly better than Trodelvy in the gastric setting, as Abizar pointed out. So the second part of the question is about ADC sequencing and why that matters. Typically, when you administer anti-PD-1 and you give an ADC, you're performing concomitant dosing. And concomitant dosing means you're giving both these agents so that there is maximum overlap between the exposures of anti-PD-1 and the ADC. And this is to make sure that it unlocks each other's efficacy. We have done experiments in urothelial bladder setting where we have given a dose-dense regimen of anti-PD-1 following the ADC treatment to see if the effect of new epitopes translates to activation. So in these experiments, anti-PD-1 did not give any efficacy on its own. The tumor growth curves were similar to control, but you get about 50% TGI where you treat with the ADC alone. But when you add anti-PD-1 following the ADC treatment, you see the curve dip. And basically, you're unleashing more activity from T cells in this setting, even though the anti-PD-1 arm by itself did nothing. So I think there is an opportunity with this payload to sequence anti-PD-1 after ADC treatment to get some kind of activity. And we have seen that in preclinical experiments.
Yeah, and I was just at a couple of points. I think that's a great explanation on the sequencing, because that data was pretty unique when we did before and after kind of regimens, and you saw a unique difference. I think the other thing I'll just add on the single agent, what also impresses me, like obviously the B cells, and we activate the B cells, that's very unique to our single agent activity. The macrophages, which are those immune system cells in the tumor microenvironment. What's interesting around cancer is that cancer is very smart. It finds a way to take those macrophages, which are typically the first responders in a sense to any kind of thing is different, and it subdues them into becoming tumor suppressive. So they actually don't do their job. The cancer kind of signals to them not to do their job. And what we found is that we actually were able to what we call repolarize those macrophages to do their job. So we got them to wake up from their trance, and the cancer cells put them in, and we found as a single agent, they got repolarized to do their job, which then starts again create activity of the immune system. That was as a single agent, and again synergizes when we had a checkpoint inhibitor. So again, we have potent single agent activity, whether it's cytotoxicity or immune activation. It just gets to really see the full light of day with the checkpoint inhibitor, as Satya mentioned.
Great. Okay, we have about 2 minutes left, so I wanted to give you the opportunity for some closing remarks before we end the webcast.
Sure. First, we'd like to thank everyone for joining the call today. Hopefully you walk away with more information around why we're excited around this novel payload, the need for a novel payload. ADCs are hot, and how we can continue to build on that and take ADCs to the next chapter and change outcomes for cancer patients. And hopefully you walk away with a clear conviction of the plan we have to get there. This is not great science for the sake of science. It's science we're going to apply to get to the clinic and make a difference for patients. And we have an accelerated plan to get there as soon as we can in a capital efficient way. So I want to thank all the participants for joining us. Thank you, Satya, for a great presentation. And thank you, JTC, for organizing and helping us today.
Great, absolutely. So this does conclude the webcast. As a reminder, the replay will be available on the company's website. We did have some additional questions, just didn't have time for them, so we will get back to everyone that submitted questions following the call. All right, thanks everyone, and have a great day.
Thank you.